Hi, my name is Amanda Podlasek, and I've been studying red tide for 5 years. This will be my 6th year in science fair study. So, my project this year consisted of 3 silent communities that looks just like this. The one that's actually inside, actually. And I was trying to find out was which location would be the best to put one of these to help the spread of red tide. So what I did was I gathered these silent communities from their locations: Sanibel location, Caloosahatchee River location, and Sanibel. And I found out in the Fort Myers Beach location that there was oysters growing here. And at the Caloosahatchee River location, I found out there was clams growing there. And the last location, the Sanibel location, I found that there was tunicates growing there. So what I did was I put each of these silent communities into 3 individual tanks, 1, 2, and 3, and then I put 13.2 cells in each of the tanks. I put red tide cells in each of the tanks, and I found out within 24 hours that all the red tide was gone. But the tunicates, they had fed the red tide within just the first hour, which was very amazing. The other 2 locations also had filter fed the water as well, but within the 6th hour, which is still very good. So from my project this year, I found out it doesn't matter what kind of location that these silent communities will be placed at, they all will filter feed red tide. Although the tunicate location does it much faster compared to the others, and it doesn't matter about the salinity. But now, this year's research, what I'm going to try to do is I did some research and I found out that there's something called the after effects of red tide. Now, you may be wondering, what's the after effect? Like, what is this? Well, the after effect is actually what hurts our health. That is when it becomes airborne and comes into our lungs. And that's what makes us cough, sneeze, gets our runny eyes. So what I'm going to try to do this year is that I already know that these silent communities can filter feed red tide. So I'm going to wait 24 hours, and the silent communities will already be placed in the tanks with the red tide in it. So then I'll wait 24 hours and then place little shrimp in the water, and then I want to see if they can live through the after effects. So the after effects would be about 3 to 4, about a week. I'm going to keep them in there for a week, and I want to see if they can survive through that, which I'm pretty sure that they can. So I have here my little paper here, and I'm just going to explain a little bit what else I'm trying to do and a little bit more detailed version of it. So my question for this year's research will be, will the silent communities help marine life survive through a red tide outbreak by preventing K. brevis populations? Now, explain my experiment is also on here. In the experiment, the researcher, myself, will gather the silent communities that were used last year and place them back at their previous locations: Fort Myers Beach location, Sanibel location, and the Caloosahatchee River after 18 months again. So after 18 months, the silent communities will be placed in 3 separate tanks, and then the red tide will be added to each of the tanks in equal quantities. Within 24 hours, the vertebrates, the shrimp, will be added to each of the tanks to see which location is the best in preventing the trouble after effects that the red tide produces. So my hypothesis this year, which I think is the most important actually in the science fair experiment, is to see, if I'm right. If I always think that's really the most exciting part of the whole experiment. Is that my hypothesis is that all locations will help with the effects of red tide. However, the shrimp in the tank with the tunicates in the Sanibel location will have healthier looking shrimp. So the way how I'll be looking for my data is by looking at the color of the shrimp. If they change their color, maybe they look a little bit thinner or a little bit more, look kind of weak. That's how I'll be getting my data. So, and hopefully everything will work out very well in this experiment, which I am pretty positive it will, and I'm pretty positive as well that it will make my hypothesis true. And I'm really excited to start working on this, but sadly I do have to wait a little bit till these selling communities are all full of filter feeders. This here is the one with the oysters, and this one, this year, I just put a big giant basket, 2 baskets on top and the bottom, and bungee corded it, because I found out at the end of last year's experiment that there was all these little fish and little crabs trying to eat the filter feeders. So I am really excited for this experiment, and I will let you know all the data and everything that's going to be going on. I'm also right now making a Google webpage where I'll be linking all of my data and graphs and everything, which you can go take a look at. Well, thank you so much for looking at this video. Bye!